pharmaphorum web editor Nicole Raleigh, and I am on site in Chicago for ASCO 2025. And with me today is Dr. Victoria Zazulina, who is Senior Vice President, Head of Development Unit Oncology at Merck. Pleasure to welcome you here today, Victoria.
Nice to meet you too, Nicole.
Now, there was a virtual media briefing to discuss key research presentations at ASCO 2025 and share updates on the company's oncology pipeline and portfolio. And pharmaphorum, i.e. myself, did indeed tune into that briefing, which you took alongside Drs. Vinod Ravi and Scott Kopetz. However, I was wondering if you could share with viewers of this video an overview of the data presented here at the Congress.
Yes, and thank you so much for the opportunity to do so, because this ASCO is actually quite eventful for us. We are bringing the results of our own work and the work of our collaborators to share with the scientific community. So some of the abstracts that probably I would like to highlight, among others, which I cannot cover all of them, are the data from pimicotinib. This is a phase 3 double-blind randomized study called Maneuver, which was conducted to assess the efficacy of pimicotinib. This is a CSF1R inhibitor, which was tested in patients with a tenosynovial giant cell tumor. This is a bit of a mouthful, but this is a real disease with a real severe consequences for, unfortunately, quite young population, because the median age is about 35-40. These are still working, active patients. And this disease is unlike many other malignant diseases, does not fit into the box of metastasizing and eventually killing the patient, but it contributes a lot to poor quality of life, limiting what they can do, pain, stiffness, impact on mobility. So this is why pimicotinib exactly was assessed, when these patients actually do not qualify for surgical intervention, which, believe it or not, so far is the mainstay of treatment in the majority of the countries. So we assessed, or our partner ASCO, in a phase 3 trial, assessed pimicotinib compared to placebo. And in this randomized trial, we saw clear improvements across all the endpoints when patients were treated with pimicotinib. After 24 weeks of receiving a daily pimicotinib, we saw that nearly all of them had shrinkage or reduction in the tumor size. Then 54% compared to 3 on placebo experienced what qualifies as RECIST response. These are the standard evaluation criteria of RECIST in oncology. And what is more important is that these patients experience, on top of shrinking the tumor, which in itself, of course, is a gratifying experience to have, they actually benefited from improved stiffness, pain, range of motion, physical performance, and that's what speaks to the patients sort of loud and clear. And we're really happy to see that both results, the primary endpoint and all the secondary clinical outcome endpoints, were moving in the same direction, very clearly signifying the benefit and the meaningfulness of what the drug can deliver.
Meaningful indeed, and brilliant, yeah, just amazing science.
Another one, if you allow me to.
No, absolutely.
Yes, yeah. So another compound which is coming more from the early stage development. This is our first ADC, which we are testing in colorectal cancer. ADC stands for antibody drug conjugates. There's plenty of them being explored at the moment. Something that we are proud of is that our program that comes from our in-house technology and platform. This is the pilot project that we took in the clinic, and already here we see very interesting results which encourage us to continue the development and, in a way, validating the whole platform to continue producing ADCs. So you will see more coming from our company. So in this particular case, we treated patients with colorectal cancer, metastatic, those who have already had a couple of lines of treatment. We selected this population because the ADC, which is in question here, it's an anti-CEACAM5 ADC. It's targeting the antigen, which is abundantly expressed in colorectal cancer. So it was almost a no-brainer to go to this indication. The payload that we deliver is a topoisomerase 1 inhibitor, quite a popular one, but all of them could slightly differ in terms of potency, in terms of the linker, which connects antibody and the payload. So all of us have our proprietary technology, and then the real differentiation comes in the clinic. So what we saw in the clinical study is that out of 60 patients that we treated across 2 doses, we randomized them in order to have 2 doses evaluated very closely and to choose which one we want to take into further pivotal development. The dose that we want to take, which is 2.8 milligram mg per kg every 3 weeks for presentabart tosemtican, that's the name of our ADC, or it was previously known under the code of M9140, we see a 31% response rate in patients, so this is tumor shrinkage sustained over time. Many patients continue in response now for months, and out of those 31 who had shrinkage qualified for response, 17% by the time when we cut the data already had confirmed response, meaning that it lasts until the next scan and the next scan, while a few more patients are still ongoing. They had their first declaration of response but still are waiting confirmation. So progression-free survival is also mapping up quite nicely, coming almost to 7 months, and this is encouraging for this patient population, also in the context of the adverse event data, which so far we can say predictably we see a chemotherapy-like hematological toxicity, which are very well known, can be managed, can be also preempted by the special care. So far so good. No cases of interstitial lung disease, no cases of ocular toxicity. Our overall database by now is more than 100 patients. So we are very encouraged to take it forward.
Encouraged indeed. I mean, it's exciting data that you're sharing with us, sharing with ASCO. So if I go back to the virtual press briefing, one thing you mentioned during that was that, and that stayed with me, was cancer is a constellation of many rare diseases, one might say. And indeed, this sentiment was seconded during an on-site press briefing the other day about how common cancers are being taken and turned into rare subgroups. So I was wondering if you could speak more to that mindset with regard to the future oncological innovation.
So indeed, this is not only a mindset, this is our reality now, developing the drugs in oncology. And if you think about what was a common features of cancer, how we think about cancer, it's the disease where cells go out of control. They have some common features and characteristics such as being able to grow, proliferate uncontrollably, invasion of surrounding tissues, and then eventually metastasizing across the body and killing a patient over a certain amount of time. Some cancers are more aggressive, some less aggressive, and can be controlled a bit better. But if you zoom further down inside the cell to understand why this is happening and on which level those abnormalities arise, with our we are much better equipped now to read the genome of those cells. And we say cancer is a disease of genome. We understand proteomics, we can see metabolomics. So everything that can characterize those cells on a much deeper molecular level. And from there, suddenly we understand that even though 2 different patients have cancers arising in the lung, molecularly, and what was the main driver turning those cells into malignant cells could be completely different between 2 patients. And this is the reality across multiple tumor types. So what is happening today, we're getting much better in understanding those drivers and developing drugs to tailor to those vulnerabilities inside the cancer cells, that in these patients we should switch off this protein. In that patients we should block that receptor, and there could be completely different pathways that we are exploring here. And I think that is why you see so many drugs now coming to the forefront, talking about specific molecular subtypes and biomarker positive groups of patients. And more you see that even our treatment guidelines, with approval of these drugs, because they do drive great response and great benefit, most of the guidelines in different diseases now say stop and think molecular testing first before you prescribe the therapy. So sooner or later, ideally, with a mindset of precision medicine, it won't be enough to say cancer of a certain organ or certain tissue. You have to really characterize whether it's HER2, whether it's RET, whether it's ALK, whether it's MSI high. So a lot of these biomarkers are emerging.
Exactly, and precision medicine also has been discussed at the conference with regard to the individual patient as well. I don't know if you wanted to state to that at all. Or...
Yes, within
Yeah.
the individual patient also, you can track and potentially predict also with AI, we get it much better. And then what would be the next step? What would be the next therapy? Because in response to in this molecular subtype, in response to our treatment, we can predict how the tumor can bypass and what could be the next resistance mechanism evolving. So in order for us to turn cancer into more chronic and manageable disease, we have to get much better in predicting those steps and having enough drugs to jump and to block those next steps before they even appear.
Yeah. So the excitement that you exude is palpable, which sort of preempts my next question. What is particularly exciting? Perhaps narrow it down to one thing if you can. Particularly exciting to you with the work that Merck is doing. Can you sum that up in a sort of summative statement?
I think it would be sort of a really genuine effort trying to use the tools and the armamentarium that we have available to us today to synergize it and cleverly combine and develop the pipeline that could build on each other, exactly thinking about precise subsets of patients and how our pipeline can address the needs, but also enhance each other in addressing those needs, but the drugs enhance each other in addressing those needs in the pipeline. So across all the approaches that we are developing, arguably all of them are precision medicine approaches that go after different patient subsets, and to unlock the power we also aim to combine between them to make it stronger.
So it preempts my final question further, which is, if you could solve one major problem in oncology that exists right now, and I know, viewers know possibly where you're going with your response, if you could solve that one major problem over the next decade, what would it be in an even more conclusional summation of what you've been discussing for us today?
So I think you think that I'm going to talk about cure, probably. And I do agree, cure is very important, but being on the side of a drug development and seeing how many failures we experience, and on the drug side, we fail the patients, we fail the drugs, we fail the patients, we fail our systems, right, by putting so much into our pipelines which eventually doesn't deliver. So one big problem I would like to resolve is to try to increase the probability of success of our drugs when they come to the clinic. And if we were from today's percentage, which is quoted is below 10% is the probability of success in oncology. If we were only to double that to 20%, I think it would be a great breakthrough, not to say even more, right, if we could do that. I think that's what could unlock, actually allow the patients already in the clinical trials, many more experience that benefit, that they don't have to wait for the drug to come to the market, and also have a much more streamlined and guaranteed path to the patients all over the world, because we would know where to go, how to design the experiments better, because we have a better feel for our drugs, and this probability of success, I think, would define also a much better use of our resources and decrease the failures, first of all the failure towards the patients, to be there right in time when they're still living with their disease and we have something meaningful to offer.
Well, thank you, Victoria, for your time today. It's been an absolute pleasure.
Thank you. Thank you.